Joel Topf with Nephrology Times. We are on the very first day of the National Kidney Foundation Spring Clinical Meeting. I'm very excited. I got Meghan Sise right here, and she has an interesting, late-breaking and high-impact clinical trial that she posted. But first of all, she's an onconephrologist here in Boston. That onconephrology field, it's blowing up. What's going on there? Why does everybody want to be an onconephrologist?
I think that's a great question. I think it's a really exciting field because there's always new drugs, there's always new kidney-related side effects that are being thrown at us. It's a collaborative field where you get to work with oncologists, and there's a lot of interest.
It really brings out the diva in the kidney, right? They're always fainting. They're always bring me to the fainting couch.
Yeah, that's right.
Okay, and always new drugs there. Why can't we get some of that energy in nephrology?
I think we are getting some of that energy in nephrology, right?
Yeah,
where there's a ton of new trials and new drugs getting approved. So
excellent. And your study today is like it's answering or starting to scratch the surface on a question that we have been just asking over and over again. Is that whenever we have to dose this, they require us to use the Cockcroft-Gault formula, and we haven't used that since Bruce Jenner was winning the Olympics. Like, what's going on? So why are we still using Cockcroft-Gault?
Yeah, this is like a real area that has always frustrated me as well, and this is why we did this study, is that, nephrologists have said for a long time, have been shouting that, Cockcroft-Gault is not a validated equation. It was developed in the 1970s. It was developed exclusively...
Before there were women, right?
Before there were women. It was developed exclusively in men. Yeah. And the correction factor for women is based on expert opinion alone. So it was never, it's a completely invalid equation.
Out line in the discussion, right?
So nephrologists say use more modern equations like CKD-EPI. And when you're making a high-risk decision, use the combined equation using creatinine and cystatin C. However, all of the oncology trials were done using Cockcroft-Gault based enrollment criteria. So oncologists say, well, this is how we did our trial, so this is how we want to dose our meds. And their concerns are
That's a pretty good argument.
Which is a great argument.
Yeah.
So when we have these conversations with oncologists, they said, well, if all of a sudden we just start using a new equation, we may be misdosing people, and we may be giving them too little chemotherapy. So there's risks on each side. If you give too much, you get toxicity. If you give too little, you might not cure cancer. So whereas both sides have really valid arguments, we knew we had to generate some data, because in my clinic, as an onconephrologist who sees patients who get chemo side effects, I was seeing a lot of women coming in with AKI and cytopenias related to their chemos. And so my hypothesis was that so many of these people are being overdosed using Cockcroft-Gault, and how can we prove that? And so what you'll see in the postdoc who works in my lab's presentation today, Dr. Siwan Kaur, he'll be presenting our work, which shows that what we did was we took everyone who's starting cisplatin or carboplatin at Mass General Hospital, and we measured a cystatin C on them before their first dose of chemotherapy. That cystatin C was not clinically available. It was research only. So the docs proceeded according to standard of care. They got a creatinine and they calculated the dose of chemo they wanted to give, and they administered it. It just so happens that we knew what their cystatin C was after the fact. So we could go back retrospectively and say, if we had known the cystatin C, how would we have dosed the chemo? And can we identify the patients who could have qualified for a dose reduction? And are those the same people who got side effects?
So those people all got dosed based on Cockcroft-Gault?
They all got standard of care, which at our healthcare center is Cockcroft-Gault based chemotherapy dosing.
Okay, but then after the fact, you knew what their estimated GFR using a combined creatinine and cystatin C.
That's exactly right.
And you found about 25% of them were misdiagnosed.
25% of them were given either a dose too high or too low.
Yeah, too high or too low.
That's exactly right. So 25% were misdosed.
And about half and half high and low.
About half and half high and low.
And the interesting one for you, or at least the one that was in your discussion, was the people that got dosed high.
Yes.
And what happened to those patients?
So if your combined equation suggested a creatinine clearance or a GFR that was lower than your creatinine clearance. If Cockcroft-Gault overestimated your GFR compared to the combined equation, you're at risk of being overdosed. So those patients who we thought were at risk of being overdosed, who received a dose that was much higher than the dose that their combined equation suggested they should have gotten, those were the patients who developed anemia, developed severe AKIs, were hospitalized for side effects. So we really think, although—
And there was a mortality signal too, right?
And there's a strong mortality signal as well.
Yeah.
So we think
this is something that people are concerned about.
Yeah, so this really sort of confirmed our hypothesis.
I'm not laughing at death. I'm not. It just looks like I'm laughing at death. It's an important side effect.
No. So it confirms our hypothesis, which is that these are our frail patients, and we might be able to identify them and potentially offer them dose reductions as a way of preventing side effects, which could help them tolerate their chemotherapy longer. And overall, that's something what we want to do next is sort of try to design studies that will test whether dose reductions can help these patients.
All right, Meghan, let's flip this over.
Yes.
I didn't see it in the results, but half those patients got underdosed.
Yeah.
Did they have worse oncologic outcomes?
They didn't have worse mortality outcomes, so they weren't more likely to die from being underdosed. And the only thing we were able to look at was if they were hospitalized for cancer progression. So they weren't hospitalized for cancer progression more, nor did they die more. So underdosing may not be as big of an issue as overdosing. We think overdosing is really where we see the problem, because within 90 days we can see those patients getting side effects, being hospitalized for side effects, and dying.
Okay, now I want to nerd out on this.
Yeah.
So one of the fundamental differences between the modern EGFR calculations and Cockcroft just gives you a creatinine clearance, and the modern calculations index it to body surface area.
So we made them equivalent.
So let's walk that. Okay, you got to where I wanted to be, but I want to walk back why that's important. Is that the reason we index it is we know intuitively that bigger people need more clearance just to keep them metabolically in homeostasis.
Yes.
But we it's just too hard for our little brains to track that. So we just want to assume everybody's the same size, and we can then compare their GFRs across populations. But that's not what when we were looking at drug clearance, that's not what we should be doing. We just want to see how fast that drug is cleared out. And so there is an option to back out that indexing, and you guys did that.
That's exactly what we did, because we wanted, I think that drugs should be dosed, and take body size and body surface area into account. So we did unindex so that they could be comparable in mils per minute to mils per minute, and when we made those comparisons.
Oh, that makes me so happy.
Yeah.
Right, but I think that's important. So when you're in your clinic at home and you're kind of talking about how am I going to use this information, remember, don't just use that simple EGFR calculation. You need to be able to unindex, and I know some of the medical calculators allow you to do that just hitting a button.
That's right. Yeah. You need to know the patient's height and weight. Yeah.
Okay, what's your take-home message?
A take-home message is that, I think that we need to study this further. We need to do trials that look at high-risk meds, and should we take into account creatinine and cystatin C? I think, our data suggests we should.
Yeah.
But, we need to study that prospectively.
So I guess today, new drugs coming on market, are they still using Cockcroft?
They are. Yeah, so many drugs in development. And if you look at clinical trials that approve new cancer therapies, the majority of them still use Cockcroft for enrollment. So I think...
I shake my head. The hematologists and oncologists, I know, they're smart people. What's wrong?
Well, I think we have to show. I think hopefully this is what our work will do is show them that they're such caring doctors. You'll never meet more caring doctors than oncologists.
No, 100%,
so I think when we show them that there's harms being done here, I'm hoping that that's what will move the needle.
I think that's awesome. Hey, this was great. Yeah. Thank you.
Thanks
Exciting
so
stuff.
much.
Hey, have you been accepted for publication yet, or
no? We actually. So we wanted to present our data as soon as possible, but we've continued enrollment. And so whereas our first abstract is 400 patients, we now have about 800 patients enrolled. So we wanted a bigger study. And so we're going to submit our paper with the full cohort enrolled.
And are you thinking you're going to go to oncology journals, or you are going to?
We want to go to oncology. That's our audience. That's where the change is going to take place. So we'll shoot for oncology journals.
Awesome. Hey, thanks a lot. This is great.
Thank you. My pleasure.